the amazing hospitality and for the opportunity to celebrate 30 years of Leptin with amazing colleagues and with all of you. So my talk is indeed on the genetics of obesity. Actually, it's like a focus on genetic variation in the leptin and also in the leptin receptor gene. So typically when we study the genetics of obesity, we focus on body mass index, because body mass index is easy to measure. It's just weight divided by height squared. And we have big sample sizes, so it means we have a lot of power. And here you see a timeline where we start in 2007 with the discovery of FTO. But then simply if we add more and more and more and more samples to the meta-analysis, we discover more and more variants. And you see here that in blue, most of the variants that have been identified for obesity have been identified for body mass index. And many of these discoveries have been led by the Icahn consortium. And I'm part of the Icahn consortium co-leading the BMI work group. And you see we haven't updated this slide for a while. I stopped here in 2021, and that's because we've been working on a new meta-analysis. This new meta-analysis includes data by the Icahn consortium, 2.1 million individuals, and we're adding data from 23andMe, another 3.5 million. So now we have a meta-analysis of 5.6 million individuals. I'm not sure we were waiting for this, or you were waiting for this, but that's sort of 5 or 6 times more individuals than before, and this is what the Manhattan plot looks like. And the threshold, genome-wide significance, is right here. So basically, apart from the centromeres, everything is associated with body mass index. Side note though, is like if you zoom in on the leptin gene, there's no variant that reach genome-wide significance. So a big challenge is, of course, the variant to function translation. How do you learn more about biology from GWASs, not only of this last one, but also of the previous ones? So that's been a major challenge. So in general, we know that genes within BMI-associated loci, if you compare them with genes outside the loci, they're enriched in the brain. That's something that's consistent with what we know from monogenic forms of obesity. But then the actual translation from variant to gene to function has been lagging. We know, for example, FTO was discovered more than 17 years ago, and we barely understand what it really does. Initially, we thought it was FTO that was the causal gene, and then later on, people thought it was a gene like next door here, RB-GRIP1L, but others thought it was IRX3. Mechanisms involved in food intake, but also energy expenditure were involved. And then, like a few years ago, there was this paper in Science that maybe they wanted to conclude the description of FTO, where they said like maybe all the genes are involved and all the mechanisms are involved. So let's move on. But in a way, this was only one gene out of the 1,000 so many loci that we've been discovered, and actually only another handful of genes have so far been followed up. So it doesn't seem that there's a lot of interest really in following up our GWAS loci into biology. Maybe one reason is because we study BMI, and BMI is a very heterogeneous phenotype, right? It's people with the same BMI have different reasons to have come to that BMI, and people with the same BMI have different profiles of comorbidity. Sorry. So in my group, but also others have tried to work on more endophenotypes, or phenotypes that lie closer to the biology. We looked at body fat percentage. We also have done multi-trait analyses where we not only look at BMI, but also BMI in the context of comorbidities. But today I want to just focus on leptin as a possible biomarker for obesity. So leptin, this is just a descriptive plot where you see circulating leptin levels measured by Olink, so a high throughput technology to measure protein levels, and its correlation with BMI, and this is what we've seen before, and there's a highly correlation. The correlation, if you express it linearly, in women in pink is 0.8 and in men it's 0.74. So in a way you could say that if you know BMI, you almost know leptin. So when we do GWASs, the genome-wide discoveries for leptin, as such, we will often, or you can imagine that you will often come across variants that are associated with both with BMI and with leptin because they are so closely correlated. So what might be more interesting is that you look at leptin levels for a given BMI. So take, for example, here all individuals have a BMI of 30, but you still see there is good variation. And some individuals, despite having a high BMI, have relatively low levels of leptin, whereas other individuals having a high BMI have really high levels of leptin. So here I would hypothesize that if you are a low leptin secretor for a given BMI, maybe you are prone to have high leptin levels in analogy with being a leptin knockout, who have no leptin and who definitely are prone to have obesity or who will have obesity. So this is another analysis that we do with the gene discovery where we look at leptin levels, imagining that everyone has the same BMI. So this is a paper that we published 8 years ago already, where we did a GWAS including data of 52,000 individuals, and we identified 6 loci. And here is the effect size of each of the leptin increasing allele within these loci. And you see FTO is part of it. And as I mentioned earlier, FTO was discovered for obesity. So the question here is like, is FTO really directly associated with leptin levels? Or is FTO associated with leptin because it is associated with high BMI, right? So if you now do an analysis where you say, like, imagine everyone has the same BMI, is FTO still associated with leptin? And actually you see it's like then it disappears. So clearly FTO is associated indirectly with leptin because it's associated with BMI. But if you do the same analysis for these other variants, actually leptin does not disappear. The association with leptin does not disappear. It's attenuated a little bit for these 4 loci, and in fact it gets a little stronger for the leptin variant. So showing that these variants really affect circulating leptin levels independent of the association with BMI. This is maybe a little small, but these are the 5 loci. So I don't show FTO because I consider FTO not to be a leptin-associated variant. So with 5 loci, each associated with BMI, and these are chromosomal regions, so it's like a snapshot from where the association happens. So here is a locus where the top variant lies next to like SLC32A1. But you see that there's multiple genes located. There's many more here, and this is the case for all these other loci. So in a follow-up analysis, we first checked whether we're trying to find the causal gene within the locus, because the nearest gene is definitely not always the causal gene. So we first looked whether these genes are expressed in adipose tissue. It's like assuming that is like critical tissue when we think about leptin secretion. And if they were, then we knocked them down in an explant knockdown model using mice. So we took the fat out of the mouse and then knocked down the gene. And we looked at gene expression and gene excretion. And then we found actually 2 loci where we indeed narrowed down that we think is the causal gene or likely causal gene. And the first one is the SLC32, where we do not think that it is SLC32 that is the causal gene, but this gene nearby, ADIG, which stands for adipogenin. And indeed, if we knock down adipogenin in this explant system, then we see that the leptin expression is down and the leptin secretion is down. And in fact, this gene had already been reported 10 years earlier by a group in Japan, and a few years later by David Savage, and Steve was also involved, indeed showing that adipogenin might indeed be involved in the leptin secretion. It is highly expressed in adipose tissue, and it is involved in adipocyte differentiation and also fat mass accrual, at least in mice. So this is actually just to show that here the variant to function translation seems much more straightforward than for leptin than we do that for BMI, because with leptin you have already assumptions about the possible mechanisms involved. Just to emphasize that GWAS for these endophenotypes might actually be more useful or informative when it comes to variant to function analysis. Then another locus where I won't go into too much detail, but there was a variant near the COBL1, and in the past, and there's also a variant associated with waist-to-hip ratio, and in the past it was believed that it was the gene next door, GR14, that was the causal gene, but then a recent study by the researchers at the Klausnister group at the Broad Institute, they really emphasized that it's again, it is COBL1, which is again involved in adipocyte biology. When we made the discovery, we did not do much about the leptin locus because we thought like, okay, we know that leptin is going to be the causal gene. It would be weird if it's another gene. But then a few years after, Olaf Dallner from Jeff's group was very intrigued by our finding because he also found that, okay, I should point out that the variant that we found was not located in leptin, but it was like located right like in the promoter region. And Olaf found in mice that also in mice there was a proximal promoter region that seemed to be regulating leptin secretion. And that coincided with our findings. What he found is like if he knocks down that region, which has a long non-coding RNA region, that the leptin levels are low in mice, and that they increase on a high-fat diet, that they increase in fat on a high-fat diet. And in fact, we more or less see the same in humans. It's like individuals who carry the variant, the leptin associated allele here that lies in that same promoter region, then we indeed see that the variant is associated with lower leptin levels, highly significantly, as you can see. The association with adult BMI is there, but it's not super significant. But what stood out in this analysis, and I think this is important, is that the leptin lowering allele is associated with higher risk of adult or childhood obesity. This in the UK Biobank, individuals who carry this leptin lowering allele are far more often reporting that they were plumper in childhood. And this is highly significant as well. So, and remember, this is not like FTO, where FTO is the variant that is associated with higher leptin, is also associated with higher BMI. Here is the leptin that is the variant that is associated with lower leptin, that is associated with higher BMI, at least in childhood. So here we assume that there's a genetic predisposition to having low leptin levels that we observe in adults, but maybe it's already present in childhood, and therefore these children may be more prone to obesity, which then carries on, but maybe gets diluted into adulthood. Then we continue doing an analysis of rare-ish variants, I should say, using an exome chip. We meta-analyzed data of 57,000 individuals, and most of these variants on that chip were coding variants. Again, we found 5 variants in this case. The most interesting one is this variant in leptin, which is only polymorphic in African ancestry individuals populations. This variant is a valiant to methionine on position 73, which is not believed to interact with the receptor per se, but structural prediction tools estimate that it might affect the stability, and indeed in some in vitro experiments we show that this methionine mutation or variation reduces leptin secretion by 20%. But what is consistent with what we observed, this methionine is indeed associated with lower leptin levels, highly significantly. The association with adult BMI is not so clear. Again, I have to remind you that here we had to do the analysis in populations of African ancestry because the variant does not exist in other ancestries. And in 4 populations, we see that in 2 populations we find an association with higher BMI. In the 2 other populations, we do not. So we are trying to figure out like what's going on. But again, what we see is a strong association with childhood BMI. And here we have data of CHOP it's a children's study in Philadelphia, and they show that carriers of this variation have the association of the variant with higher leptin is particularly pronounced in the first few years in life. It's almost like putting kids on a higher track in life. And once they are age 7, they're on that track, but then the leptin variant does not seem to have an effect anymore. But then the kids are on that track, right? Maybe on a track for obesity. And again, here we can assume that the variant that's associated with adult lower leptin levels is not unlikely to be associated with lower leptin levels in childhood, and that may be the cause of the higher BMI in adulthood. Again, similar, but maybe in a softer or weaker version of the leptin knockouts. So this is all data from 8 years ago, and since then, more leptin level data has become available thanks to these high throughput technologies such as Olink. So we've taken advantage of that, and actually we're now performing meta-analyses using all the data that's available in the public domain, and sometimes also approaching groups. It takes some time to harmonize all the data and make sure everything is on the same scales. So now I'm going to present some data of meta-analyses of the UK Biobank with our previous analyses, and we now have a sample size of 71,000 individuals, so 50% larger analyses. We replicate all the previous findings and find a few more loci. If we adjust, so we have 14 loci here. If we adjust the analyses for BMI, then FTO again disappears, this SOX1 disappears, and this olfactory one disappears, but we add a few more loci. We need much more time to do the variant to function translation here. But these are the effect sizes of these 20 loci. So each variant here is shown with the gene named here. You see the variant here with SOS2 stands out. It's a rarish variant located in the gene. It's a coding variant. And I can actually not figure out why that variant or that gene in particular would be involved in leptin secretion. The only thing I can find is that this gene is involved in the Noonan syndrome 9. If we adjust for BMI, you see that many of the associations with leptin survive the adjustment, so showing that these variants are associated with leptin level irrespective of BMI. 3 of the variants are located in the leptin gene or nearby, or in the leptin locus, I should say. And then these 5 loci are located in this COBLL1 locus. If we now make a score of all these leptin variants, so the score means that we bring together all the associations with leptin in one score, or with leptin adjusted for BMI in another score, then people with a high score will have high leptin levels for one score, or with the other score will have high leptin levels irrespective of your BMI. And then we test association with various phenotypes. So for example, if you take the score unadjusted for BMI, then you see indeed that if you have a high score for leptin levels, then you have a high BMI. It's like no surprise. But if you then adjust, or if you use the adjusted score, then the association with BMI almost by definition disappears. That's what you see here. And most associations with all these anthropometric phenotypes sort of regress to the null. There seems to be some effect on fat distribution, but we're still looking into that. Maybe it's because we adjust for BMI and not for fat mass. We'll come back to that another time, I think. But we do see, what I think is the most remarkable result again, is that if you look at the red dot here, this is association between, I would say, innate leptin levels, irrespective of BMI. So if you have a high score for innate leptin levels of leptin, if you have a high score for innate leptin levels, then it seems that in childhood you report more often to be thinner. So again, in analogy with the leptin knockout phenotypes. Just a few more words on the leptin receptor. We measure leptin, circulating leptin receptor, soluble leptin receptor. It's very different from, or it is different from the leptin receptor in the brain. Leptin receptor gene encodes 5 isoforms as a result of alternative splicing, and 4 isoforms have this membrane region, whereas the soluble leptin receptor that we measure does not have that. And it is believed that the soluble leptin receptor binds to leptin, maybe to help delay the clearance of leptin. Phenotypically, we see that there is somewhat a negative correlation between leptin receptor levels and body mass index, and it seemed the amount seemed to be similar in men and in women, and the correlation with leptin levels, so circulating leptin levels on the y-axis and leptin levels is also negative, also because leptin levels and BMI are highly correlated. So it's a negative correlation, but definitely not as strong as leptin and BMI. So also, now 14 years ago already, there was one small GWAS study in Nurses' Health, and they did a GWAS on leptin receptor. And what they found is like one hit, and that was indeed the leptin receptor. In fact, within that locus, there were multiple signals. So now we did a meta-analysis, or we did analysis using UK Biobank data, and we have 39,000 individuals, like many more. And you see more loci, but again, leptin receptor, as you can see here, stands out. The PDE4B is located in that same region, but adjustment actually for this locus does not make, adjusting for BMI does not make a big difference here. These are the 50 loci that have been identified, adjusted for BMI here in orange. Most of these, or many of these independently associated variants are in the leptin receptor gene. Several of these are also in this GCKR locus, which is a mystery locus, if you ask me, because it seems to be associated with any phenotype. But then there's still another series of loci that seem to affect leptin levels irrespective of their association with BMI. This is of course preliminary data. We're still very much looking into like, what might be the causal genes within each of these loci. Again, if we make a leptin score, the associations with these anthropometric phenotypes are far less pronounced as with leptin, as with a leptin score. If you adjust for BMI, everything sort of attenuates to the null line, so meaning that we actually don't see an association between a genetic predisposition to high or low levels of leptin receptor, soluble leptin receptor, except maybe again for this association with adult BMI. But that could also be just a reflection of leptin receptors association with leptin, but still intriguing. So this is, there's a lot more to come on this story now that we have new data on leptin levels and on leptin receptors, so more is to come, but I want to conclude with the GWAS for circulating leptin and leptin receptor does yield new loci, often independent of BMI, makes it interesting. A genetic predisposition to low leptin levels seem to predispose to childhood obesity, and I think that is very intriguing because it is consistent with the leptin knockout populations, and maybe it is a type 1 or a type 1.5. And maybe if we talk about precision medicine, these are the kids, or if we can measure their genetic score at birth, maybe these are the kids to keep an eye on, and maybe we should they could benefit from leptin. I'm not a clinician, so I don't want to go that far. We are now working on measuring leptin at birth, at 6 months, at 18 months, at 6 years, to really see like, what's happening during these very early years in life. So hopefully more is to come from that type of analysis. And we're also will be working on the variant to function analyses after we meta-analyze all the data, so that we don't have to do any more meta-analysis after that. I'm grateful for people in my team in New York, but also in Copenhagen, the Giant Consortium, Jeff's collaboration with Rudy Hanye at the Lincoln University. Particularly want to highlight City's efforts in the last few weeks to help me do these meta-analyses. I'll finish here. I've been asked to put this up. I'm organizing a Keystone. Jeff will be there. So if you listen to his talk again, please come. I'm happy to take some questions.
Thank you, Professor Loos. It was a great talk. And I think we can